Hello students, Mr. Kirk here, educator with the Meadowlands Environment Center. Some of you may have met me before here at the Environment Center, or if I came to your school with our mobile food lab. Maybe not. Either way, today I'm doing something a little different. I'm presenting this, the very first episode of Finding Science, our video series for the science-minded individual. Now you might be asking yourself, wait a minute, is that me? Am I a science-minded individual? Well, the answer to that is probably because lots of people are science-minded. Being science-minded doesn't mean you're a scientist. It just means that you're willing to use science. Now, what do we use science for? Whether we're a scientist or not, we use science to answer questions, to solve problems, and to understand the world better. Pretty important stuff, right? So you might ask, Mr. Kirk, why would anyone not want to be science-minded? Why would they not want to use science, especially if it's going to help them do all of those things? Well, I think that's a very good question. And I think the answer is science can be difficult. Even though science will often give us the answer to a problem we're looking for, it may take a very long time. And in fact, the investigation that we're going to be doing today is a really good example of that. Now, the investigation itself doesn't take that long, but what it's going to show us is something that scientists were looking for many, many years. Now, there are a couple of reasons why it took many years, but one of the reasons is that they got the wrong answer many times. And I've even heard a scientist say that if you want to be a scientist, you have to be willing to get the wrong answer many times before you get the right answer. If you're not willing to do that, then chances are you're not going to be a scientist for very long. Now again, you don't have to be a scientist. We're just asking you to be science-minded. Okay? Do your best to use science, and that's going to be the best way that you can find the answers to so many questions. So if you're willing to do that, we are ready to move ahead with our first investigation. All right, everybody, so now you know what it is we're doing on Finding Science today. It's an investigation known as a DNA extraction. And I think it's important that we talk a little bit about those terms before we go any further, just so we have a better understanding of what it is we're doing. So I'm going to start with DNA. That's not a word, it's 3 letters, DNA. And that means it's an abbreviation. I know you know about abbreviations. You can probably think of one right now. The one that I think of first is USA, which of course is short for United States of America. So DNA is short for deoxyribonucleic acid. That's 2 words, only 2, believe it or not. Deoxyribonucleic is the first one and acid is the second one. So deoxyribonucleic acid. Now, that is quite a mouthful. It's not terribly important that you're able to say those words right now. It's nifty if you can. So maybe later you want to practice and you'll be able to say those. For now, we can just say DNA. Now the second word, extraction. That word describes what it is we're actually doing with the DNA. Think about a tooth extraction. Maybe some of you have heard of that. Hopefully you've never had it done because it's pretty painful, but that's when you have a tooth pulled out of your mouth. In a similar way, a DNA extraction is when we take the DNA out of where it is, okay? Now the next question is, where is the DNA? Well, DNA is in every living thing, okay? So that means I have DNA in me, you have DNA in you, everything has DNA, everything that's living has DNA in it. Now, we could actually take DNA out of ourselves. In fact, most of us have had DNA taken out of us. If we've been to the doctor and we had a blood sample taken, that blood has lots and lots of DNA in it. So we could do that. It takes some training though. So instead, we're just going to take DNA out of something else. It is something that was living at one time, but it's not ourselves. So we're going to take a look at that next. All right, everybody, so let's take a look at what we are going to use for our DNA extraction today. I've got a couple back here. Maybe you have some of these in your own home, since these are a tasty treat for a lot of us. That's a strawberry, right? And you know that a strawberry grows on a plant. Maybe you've been to a strawberry farm and picked your own. That's a lot of fun, right? And you know that once you pick that strawberry from the plant, it's not getting the water, it's not getting the nutrients that the plant provides, so it's not really alive. But even though that's true, so long as the strawberry looks like a strawberry, it's still going to have DNA in it that we can take out, that we can extract. And in fact, if we preserve the strawberry for a while, maybe we put it into the freezer, that's going to keep the strawberry looking like a strawberry for a very long time. And that means that the DNA is going to stay intact. It's going to stay in one piece, and we can extract it long after we put it in the freezer and learn about the strawberry from the DNA. What's really interesting is we can find an organism that's been preserved for thousands of years, and there's a very good chance that that DNA is still intact and we can extract the DNA from that organism, maybe a woolly mammoth or something like that, and we can use that DNA to learn about that organism. Now let's go back to the strawberry and how we know it's a strawberry. Of course, we can smell the strawberry, we can taste the strawberry. Those things are going to tell us for sure that it's a strawberry, but also we can look at it and know that it's a strawberry because it's red, it's got a certain shape, right? It's big at the bottom and smaller at the top. It's got these interesting leaf-like structures on the top there, which aren't really leaves. We can maybe talk about that a little bit later on, but the point is there are things that we can see and smell and taste about this organism and know that it's a strawberry based on that information. And that information is known as physical characteristics. What we see and smell and taste about something are its physical characteristics. Now, remember a moment ago I said we could learn about an organism from its DNA. We could learn about the strawberry, we can learn about the woolly mammoth. What we're learning is the physical characteristics. Now, again, we can determine those by looking or sort of interacting with the organism with our senses, and that's fine. But we can also learn about those physical characteristics using the DNA. Now, the challenge is, for us, we can't see that information in the DNA. We are going to see DNA today, but if we use powerful science tools, we can see more of that DNA. It's just like using a microscope to see more of a small living thing, right? So what we're learning about when we learn about DNA is really the physical characteristics of an organism. So we were just talking about the physical characteristics of a strawberry, right? The way that it looks, the way it smells and tastes. Remember, those are things we already know about the strawberry, and being science-minded is about asking questions that we don't know the answer to about something, right? So just as an example, a question you might have about the strawberry is, how long does the fruit grow on the plant before it's ready to be eaten? Now, of course, that's not something that we can investigate now. That takes a little while, but it is something you could do on your own. You could take some strawberry seeds and plant them, watch the plant grow. Once the fruit comes out, you can see how long it grows before it's ready to be eaten. So I have a silly question for you now. Let's imagine that we want to build a strawberry. And I don't mean grow the strawberry plant, we just talked about that. I mean we actually want to build a strawberry. Okay? And I don't mean one like this. Of course, we could build a strawberry out of plastic building blocks, and it's going to look a little bit like a strawberry. It's kind of neat, but it's certainly not going to feel like a strawberry. It's not going to smell or taste like a strawberry. So what if we wanted to actually build a real strawberry? Well, we can't do that. We don't have magical powers, but imagine that we did have magical powers. Where would we start? We'd have to start with something called a cell. The cell is the smallest part of the strawberry, and you have cells inside of you too. Of course, they're not strawberry cells unless you eat the strawberry, but your cells are different than a strawberry cell, and you have lots of them. And you've probably heard of these. You have muscle cells, like heart cells, you have skin cells, you have neurons, which are brain cells, you have so many different cells and so many in number. Okay, there are more cells in your body than there are people on the entire planet Earth, right? So what's important about those cells is they are what contain the DNA. Okay, the DNA is in the cell. It's not just floating around in your body. It's in the cell. And that DNA is actually what builds the strawberry and builds your body. Okay? If we were going to build a model of the strawberry, like you saw a minute ago, we would need some instructions to do that. That's what the DNA is. Okay? The DNA is instructions that tell the cells in your body to make more cells that look just like them. Okay, I'm going to go over those supplies you saw listed there just a minute ago to give you some information about some of them, as well as substitutions for one or two in case there's something you don't have at home or you're just not able to find it in the store. And I'm going to start with the strawberries. Now remember that all living things have DNA in them, so if you don't have strawberries, you actually could use blueberries or bananas for the extraction, and those usually work pretty well. We do find the best results with strawberries, and that's probably because they have more DNA packed into their cells than the cells of other organisms. But again, if you don't have strawberries, you could try blueberries or bananas and hopefully get the same results. Something to remember about whatever fruit you're using is that it doesn't have to be in perfect condition. If it's a few days past the point when you'd actually want to eat it, kind of got lost in the back of the refrigerator there or whatever, that's okay. If you're using strawberries, for example, and they're a little moldy, if you don't mind, you can just scrape that off of there and still use them. It's not going to affect the extraction at all. So that's something to keep in mind. Okay, you need a half a cup of water. Okay, this can be regular tap water. It doesn't have to be anything special as long as it's clear. Half a cup of water. You need some rubbing alcohol. Okay, this is something that we have here at the Science Center. Most people have it in their homes. If you don't, it's usually pretty easy to find in the health and beauty department of a grocery store or a convenience store or something like that, a department store. So hopefully you can get your hands on that. That should be chilled. I have it sitting on ice right here. You can put it in the freezer. It will not freeze. You can put it in there for an hour overnight, however long you want. But an hour is sufficient. You need some dish detergent, and hopefully you have some of this. If you use an automatic dishwasher and you don't have any of this, you can use shampoo or hand soap, liquid hand soap, that works. All right, you need 3 cups. Okay, I use plastic so I don't have to worry about breaking it, and I like the transparent because the ones that are see-through, because then you can see the DNA start to separate at the end of the extraction. Right through the side. You can also see that through the top. So it's not terribly important, but it is kind of nice to have at least one for the end that's see-through. You need some table salt. Okay, some table salt, regular old table salt, nothing special about that. Okay, you'll need a coffee filter or something to filter the DNA out at the end of the extraction. If you don't have a coffee filter like this, if you have the cone shape, that works perfectly well. You can also use paper towel if you don't have a filter. A zipper seal bag. This is the size that I like. It's just about perfect for 2 or 3 strawberries. You can use a slightly smaller one, the half-size snack size zipper bag, or a larger one, of course. It doesn't really matter terribly as long as it's a zipper seal. And you need a wooden thing like this. This is a skewer, of course, but you could use a popsicle stick or craft stick, something like that, just to scoop the DNA out at the end of the extraction. So those are all the supplies that you need. Hopefully you can get your hands on all of that. And once you have it all together, then we are ready to proceed with the extraction. What we're doing in this step is removing the leaf-like structures at the top of the strawberries. We do this because the DNA, remember, is inside the cell of the strawberry, and because these leaf-like structures are very tough, it's difficult to break them apart to get to the cells, to get to the DNA, so we just pluck them off and put them aside. Once you've removed the greens from the strawberries, the next step is to place the strawberries into your zipper sealed bag for mashing. It's important before you seal the bag completely to gently press out as much of the air as possible. This will prevent any large air pockets from forming, which will keep you from popping the bag as you mash the strawberries. You can see we've sped the video up here quite a bit just to save time, but to thoroughly mash the strawberries, you want to do this for about 2 minutes. And what we're actually doing in this stage is we are separating the cells of the strawberries. The next step is actually going to break apart the cells to get to the DNA. This is just getting us ready for that. All right, we've done a nice job mashing up the strawberries. That was a lot of fun. And that helps to separate the cells of the strawberry, remember, but now we need something to break the cells open so we can get to the DNA inside. So we're going to make up a liquid now using water, table salt, and dish detergent. We have a measuring spoon for measuring and mixing, and a cup to hold everything together. We're going to start with water. This is 1/2 cup. Remember, this can be tap water. And this is a solvent, which means it's going to dissolve the other two ingredients. It will allow all the ingredients to mix together and make contact with all the strawberry mush so we get as much DNA as possible. We're going to add to that 1 teaspoon of salt. And we'll add to that 2 teaspoons of dish soap. And this can be, remember, hand soap, liquid hand soap, or shampoo if you don't have any dish detergent. And this reminds us why it is so important to wash our hands. This is really the ingredient that breaks open the cell of the strawberry. And if you have viruses on your hands, for example, washing your hands vigorously with soap will help to break apart that virus and kill it. So we're mixing our ingredients a little bit. We don't want to be too vigorous here because we don't want too much sudsing. But we do want those ingredients to dissolve thoroughly. Now we're going to add this, some of it. 3 teaspoons is usually good. We add that to our mashed strawberries. And if you need somebody to help hold the bag up and hold it open, that's always helpful. So again, this is going to really start to break open the cells of the strawberry so we can get to that DNA. Now, of course, we want to make sure that bag is nice and sealed, and just like before when we were mashing the strawberries, we want to squeeze all that air out of there. And then you want to mix up the DNA extraction liquid with the strawberry mush. We've sped this up, of course, but you should do this for about a minute. All right, after a minute or so of mixing the DNA extraction liquid with the strawberry mush, you want to remove the solid bits from the mixture using the filter. Carefully push the filter partway into the cup, folding the wavy edge over the top. You'll see here I'm squeezing or sort of crimping the filter where it rests on the cup so it doesn't fall in. If you have a helper, you might want to ask this person to hold the filter during this step to prevent any mishap. Now you want to pour as much of the strawberry mush into the filter as possible so you can get as much DNA as possible. Now after a few seconds, you'll see a little bit of strawberry juice start to drip through the filter. And this, of course, is where a lot of the DNA is, even though we can't see it yet. You want to wait about 5 minutes. You can wait a little longer, but at least 5 minutes for most of the juice to drip down into the cup. And then it's really just one step after that until we get to see some real DNA. I can't wait. After 5 to 10 minutes, you should have about a tablespoon of liquid in your cup, maybe a little more, and that's enough to complete the investigation. But to get the most DNA, you want to try what I'm showing here. Bunch up the edge of the filter and gently squeeze. Keep in mind, you want to be very gentle here. If you squeeze too hard, you might break the filter, which means you'll have to repeat this step. Now, it's difficult to imagine just how much DNA is coming through the filter and even just a single drop of that strawberry juice. But keep in mind, a human hair, which is really thin, is 60,000 times thicker than a strand of human DNA. So even though we can't see these individual strands of DNA, we know there is an awful lot there. All right, we've reached the final step before we get to see some DNA, and that step is to add an amount of rubbing alcohol equal to the amount of strawberry juice that we have in our cup. So we want to put that rubbing alcohol into a separate cup first to be sure we don't add too much. We don't want to waste it. And then we just go ahead and pour that into our strawberry juice. And pretty soon after that's in there, we should start to see the DNA separate itself. The alcohol helps that clump together. It helps the DNA clump together. So pretty soon we should see some separating, and I can see a little bit there already. All right, after 5 minutes or less, most of the DNA will have clumped together, so it's time to take a look. So you need to just tilt the cup to the side and get in there, scoop up that white substance. There you have it, strawberry DNA. Now remember, part of being science-minded is using your senses to observe, right? So it's okay to touch it. It's not anything that's going to hurt you, of course. You don't have this DNA in your body, but you have human DNA, so it's certainly not harmful. It does feel quite interesting. All right, students, we have done it. We have successfully extracted DNA from a couple of strawberries. Amazing stuff. If you had the same results that I had, you ended up with something that looks a little bit like snot. I know that sounds kind of gross. I don't think it's improper, though. It is a really good description of the way this looks. Of course, we want to remember that that's not snot. It is DNA from the cells of the strawberry. And also, it's important to remember that that is the instructions for the cells. So for the strawberry cells to make more cells, they need that DNA. For your body's cells to make more of themselves, they need that DNA as the instruction manual. Just like we said, you need the instructions to build a model strawberry or whatever, out of plastic building blocks. Now, there's not a whole lot that we can do with this DNA. I mean, we could preserve it if we want to. We need some pretty high-tech equipment to do anything with it at this point. But in the real world, of course, they have that high-tech equipment. And so forensic scientists can use that DNA to find the criminal that committed the crime. If they have a sample of their blood or something else that has DNA in it at the crime scene, they can connect that to the criminal. If a person is sick, then doctors and nurses can look for the DNA of foreign organisms in their body, and that will help them determine what sickness, what illness the person has. So DNA is important for us, of course. It helps us grow, helps us be alive, but it's actually useful in the real world in many different ways. So I hope you enjoyed the DNA extraction and you learned a little bit. And I hope you'll tune in the future, of course, to the next episode of Finding Science. See you next time. Well, if you're still here, you're probably looking for an explanation about why in the video I called the top of the strawberry the bottom and the bottom of the strawberry the top. So, here goes. This is a picture of a strawberry plant with lots of strawberries on it. And what's different, of course, about all these strawberries is that they're at different stages of development. Strawberry number 2, for example, hardly even looks like a strawberry. It's pretty small, and it's kind of greenish in color. Strawberry number 3 certainly looks more like a strawberry. It's a lot bigger, and it has sort of a pinkish color to it. Strawberry number 4 is one that is just about ready to be picked and eaten. Now, what is number 1? Well, number 1, of course, is the strawberry flower before it becomes a strawberry. Once that flower is fertilized, the center of it will develop into the strawberry. What we will see first when that happens is the bottom of the strawberry. When that strawberry gets big enough, the flower will start to droop. And guess what? The bottom of the strawberry becomes the top, and the top of the strawberry becomes the bottom. So that's my explanation. The top of the strawberry is both the top and the bottom bottom of the strawberry is both the bottom and the top. Thanks for listening.